Hi class, this is Dr. O'Connor. In this video, we're going to talk about enzymes. We're on chapter 19, enzymes and vitamins. We'll talk about both. So what are enzymes? Enzymes are biological catalysts, and we talked about catalysts last semester. And we know that a catalyst is used to speed up a reaction. So if we look at the graph here, we have two curves. We have the blue curve here that shows a reaction progress without a catalyst. And then we have this green curve that shows the reaction rate with a catalyst. And remember, a catalyst lowers the activation energy. Okay? That's all it does. It doesn't affect the energy of the reactants or the products. It just lowers that activation energy. And it does not affect equilibrium. We might get there a bit faster with a catalyst, but it's not going to affect the position of equilibrium, if you will. So, a little review here. Here we have our reactants, the reactant progress going from reactants to products. At this maximum here, so from here to here, that's the activation energy, and that's for the uncatalyzed reaction. And for the catalyzed reaction, we see the activation energy is lowered. So a catalyst, it provides an alternate pathway for the reaction to occur. So the reaction will be faster, and it's faster because the activation energy is lowered. There are so many chemical reactions that take place in the cells. There are 2,000 different specialized proteins called enzymes in each cell, and each one is used for a different reaction. Enzymes are highly selective, and what we mean by that is they will only act as catalysts in certain chemical reactions. So let's do a few definitions here first. So an enzyme, a protein or other molecule that acts as a catalyst for a chemical reaction. Enzymes are catalysts for biological reactions. A catalyst will decrease the time it takes to reach equilibrium by decreasing the activation energy. And biological enzymes tend to be water-soluble globular proteins. That's because they have to travel through the aqueous body fluids. An enzyme will have a pocket within the structure that is called the active site. And this active site has a specific shape and all of the chemicals that are necessary in order to bind or act upon a molecule. We call that molecule that's undergoing a transformation a substrate. So this is the reactant in an enzyme catalyzed reaction. So you can think of the active site as a little chemical laboratory that has all of the necessary chemicals that are needed in order to transform that substrate. As I said before, the activity of an enzyme is limited to a certain substrate and a certain type of reaction. And this is referred to as the specificity of the enzymes. And enzymes will differ greatly in their specificity. And we'll go through some examples here in a moment. Enzymes are also specific with respect to stereochemistry. So a particular enzyme might only catalyze a reaction of only one enantiomer. So for example, this figure here, this is a representation of the enzyme lactate dehydrogenase. And what this enzyme does, it catalyzes the removal of a hydrogen from L-lactate. Okay, but it will not remove the hydrogen from D-lactate. So in other words, this active site is very specific for the L isomer. The L isomer will fit into the active site, whereas the D isomer will not, and therefore no reaction will take place. We measure the catalytic activity of an enzyme by its turnover number. And the turnover number refers to the maximum number of substrate molecules that are acted upon by one molecule of enzyme per second. The number of substrate molecules that can be transformed, shall we say, by one molecule of enzyme per second. So let's take a look at this table here. One example is catalase, and this protects against molecular damage that's caused by free radicals. And catalase can turn over more than 10 million molecules per second. Its turnover number is 10 million. So let's take a look here. Ribonuclease, this catalyzes the hydrolysis of phosphate ester links in ribonucleic acid, or RNA. Its turnover number is 100. Okay, the turnover number is very important. This one here, this papain, this is catalyzes the hydrolysis of peptide bonds, only 10 molecules a second. So we see that different enzymes have different turnover numbers, and there's a huge range of turnover numbers. Let's talk a little bit about enzyme cofactors. A cofactor is a non-protein part of an enzyme that is essential to the enzyme's catalytic activity. And a cofactor can be a metal ion. Oh, could that be why we have to take minerals? All right, we need zinc and metals like that. So it could be a metal ion or what we call a coenzyme. Many enzymes, not all, are conjugated proteins that have these non-protein portions, and we call them cofactors. So a metal ion or a coenzyme is a cofactor. Some are metal ions, others are non-protein organic molecules. The non-protein organic molecules are called coenzymes. Here are some of the inorganic ion cofactors: copper 2, iron 2 or iron 3, potassium. We have magnesium, manganese, molybdenum, nickel, selenium. Okay, selenium and zinc are both found in very, very small amounts. We call those trace minerals, if you will. So, in order for an enzyme to be active, it may or may not require a metal ion, a coenzyme, or both. Some of these cofactors are held tightly by the enzyme by non-covalent attractions, or they might be covalently bonded to the enzyme. Others can be loosely bound, and then this way they can leave and enter the active site as they're needed by the enzyme. So they're not permanently with the enzyme, only when the enzyme needs them in order to catalyze a reaction. Cofactors provide additional chemically active functional groups which aren't present in the active site. Remember, if we have a protein, right, that active site is going to be composed of those amino acid side chains. So there are some reactions that need other functional groups besides those side chain functional groups. This is why cofactors are needed. Some of the metal ion cofactors are iron, zinc, copper, manganese, molybdenum, cobalt, nickel, vanadium, and selenium. Metal ions may anchor a substrate in the active site, or they might participate in the catalyzed reaction. It just depends on the reaction. And there are so many of them, over 2,000 of these types of reactions in each cell. Here's an example here. Here we have an active site, And in that active site, we have a histidine, actually 2 histidines, and this glutamate. And here, within this active site, we have a metal ion, the zinc ion. And this zinc ion is covalently bonded to the nitrogen, oxygen, and this nitrogen here. And these are coordinate covalent bonds. And why is that? That's because the nitrogen and the histidine and this oxygen here are donating the pair of electrons for the covalent bond. So back in Chemistry 251, I told you that some of the, especially the transition elements, they're able to form covalent bonds, coordinate covalent bonds. So in this case here, in order for a particular enzyme to work, you might find a metal ion in the active site, and it'll be bonded to these atoms, usually nitrogen or oxygen, by coordinate covalent bonds. Here we're going to classify the enzyme. There are 6 classes of enzymes, so 6 classes of enzymes. And I want you to look over this table 19.4 in the book. I will have questions about these classes of enzymes. And here we have the first class that we'll talk about is the oxidoreductases. And these enzymes catalyze oxidation reduction reactions. And we can see oxy, right, and reduct. That's both in the name. And the ACE, ASE, is usually a suffix for most enzymes. You'll see that suffix, ACE. Some of the examples, alcohol dehydrogenase is found in liver cells. It can oxidize naturally occurring alcohols found in foods to aldehydes and ketones. In fact, methanol, as we know, is a very toxic alcohol. And in the body, the reason it's so toxic to us is because it is oxidized to formaldehyde in the body. Okay? So, again, oxidoreductases catalyze oxidation reduction reactions. And here are some of the subclasses. I won't go into those. You can read those. Transferases, these are enzymes that catalyze the transfer of a functional group between 2 different compounds. The subclasses, transaminases, we'll talk about those later, and kinases. And we will talk about those later. So in this reaction here, we haven't talked about ATP yet, but basically what this is a transfer of a phosphate group from ATP to this fructose 6 phosphate to produce fructose 1,6 biphosphate. But basically it's just a transfer of a functional group. In this case that was a phosphate group. Hydrolases, these catalyze bond breaking with the addition of water as hydrogen and hydroxyl group to the fragments. We've seen these before. And remember, the chemical reactions that we looked at for small organic molecules are basically the same for these larger molecules because of the functional group. Here we have some subclasses, and here is an example. Here we have this polypeptide, okay? And basically what's going to happen is we have a protease enzyme, and proteases break proteins into peptides and amino acids. So we see that this bond here is going to be cleaved, and we end up with a shortened polypeptide and amino acid. So hydrolases, a hydrolase, in this case more specifically a protease, is needed for this particular reaction. Lipases break glycerides into glycerol and fatty acids. We'll talk about those later. Amyloses break starch into sugars. We know about the starch amylose. And nucleases break deoxyribonucleic acids, DNA and RNA, into nucleic acids. The next class is an isomerase, and this catalyzes the rearrangement of atoms in a substrate. So for example, here An isomerase is used to catalyze the rearrangement of the atoms in the dihydroxyacetone phosphate. And when these are rearranged, notice here was a carbon oxygen double bond, okay, but see what happens here. Now the carbon oxygen double bond is located here, and here's a carbon bonded to an OH group. It's going from trans to cis or cis to trans. All right, that would also be another example. Here we have the liases, and we have the subclasses. Decarboxylases, oh, what would that be? Catalyzes the removal of a carbon dioxide. Deaminase catalyzes the removal of an NH3 group. Dehydratases catalyze the removal of water, and hydratases catalyze the addition of water. So basically these catalyze the addition or elimination of a functional group from a substrate without a hydrolysis reaction. So here we see fumarate, okay, it undergoes this reaction. Fumarase is the catalyst, the enzyme catalyst, and so fumarate is, notice that a molecule of water is added to the fumarate and it becomes L-malate. We're going to look at this later when we get to the citric acid cycle. Ligases catalyze the bonding of two substrate molecules. So, for example, RNA, DNA, and protein, we can think of those as biological polymers. For example, proteins. We know that proteins are many amino acids that are linked together. So these ligases catalyze the bonding of substrate molecules in order to maybe form these larger molecules like proteins, DNA, and RNA. So take a look at these, and you can look at them in more detail. I just wanted to point them out here. Okay, how do enzymes work? Well, why are enzymes specific? Why do they have specificity? And where is it found? The specificity is due to the active site. Remember what I said, the active site is that pocket in the enzyme that has the specific shape and the chemicals or chemistry, chemical makeup, if you will, in order to react with a specific substrate. So this is found in the active site. So what this means is that enzymes specifically react with only one or maybe a few similar compounds that are similar in shape, if you will. So we have two models that describe the interaction between substrates and enzymes. The first one is the lock and key model. So we can describe the substrate as being a key that fits into the active site, which is the lock. And so we have the enzyme being the lock and the substrate is the key. And just like your front door, only the correct size key is going to fit into the lock. So what that means is only the correct sized or shaped substrate is going to fit into the active site of the enzyme. So we think of this as being, the enzyme as being very rigid, at that active site, very rigid, the same with the substrate. So we have this rigid lock going into this rigid active site, or rigid key going into this rigid active site. But actually, this second model explains this a lot better. And this is a model of enzyme action in which the enzyme has a flexible active site and that can change shape in order to fit the substrate and then catalyze the reaction. So basically, you have the enzyme and the substrate that are approaching one another. And because of the interaction between the substrate and the active site, then the right fit between the two is induced, and then the reaction can be catalyzed. And so what are we talking about? Interactions between the substrate and the enzyme. Remember, in the active site, you have those amino acid side chains, and so that's going to be important as to whether the substrate will navigate to that active site. Okay, so let's take a look at what happens here. We have a substrate that migrates to the active site, and the goal is to form an enzyme substrate complex. So the substrate is drawn into position by non-covalent forces, with enzymes within that enzyme substrate complex. Atoms that are going to form new bonds will connect with one another. Bonds could be with another substrate, or there could even be temporary bonds with atoms in the enzyme. And don't forget, we also have any cofactors that might be needed for the reaction. Groups that are needed for catalysis have got to be close to specific locations on the substrate. Remember, some molecules might be too small to induce the proper alignment, and therefore will not be able to react. Only the proper substrate is going to be capable of inducing the proper alignment of that active site. Remember, here I said groups for catalysis must be close to specific locations on the substrate. Many reactions require acidic, basic conditions, or metal ion catalyst. The active site on the enzyme can provide these conditions without disrupting the pH of body fluids. The necessary metal ions, remember, are present as cofactors. So once the reaction is complete, the enzyme and product molecule will separate, and then the enzyme will restore itself to its initial condition, and it's all ready to participate in a reaction with another substrate. Enzymes act as catalysts. Why? Well, number one, because of this proximity effect. And what is that? That's because enzymes can bring the substrate and catalytic sites together. So we talked about the interactions between substrate and catalytic sites. There's the orientation effect. Remember, we talked about this back in Chemistry 251. There has to be, in order for a reaction to take place, orientation has to be just right. So enzymes act as catalysts because of their ability to hold substrate at the right distance and in the correct orientation needed for the reaction. Also, they can provide acidic, basic, or other types of groups that are needed for catalysis. This is the catalytic effect. And they can lower the energy barrier by inducing strain and bonds in substrate molecules. This is the energy effect. So basically what happens is, remember, you need a lot of energy to break bonds. So as the active site, because we have a flexible active site, then the bonds are just distorted. So that means it's going to take less energy to distort the bonds rather than break the bonds. So that means less energy. So the energy is lowered, that energy barrier, remember, that activation energy, that is lowered because strain is induced in the bonds in the substrate molecule rather than the bonds being broken. Let's go on now and talk about enzyme activity and concentration. And if we were to plot reaction rate and as a function of substrate concentration, what we see is at low substrate concentration, the reaction rate is directly proportional to the substrate concentration. So here, low substrate concentration. So in this range here, we kind of have a linear relationship. As the substrate concentration is increased, though, we see that the rate tends to drop off. So as we increase the substrate concentration now, rather than going like this, we see the rate starting to drop off as more of the active sites are occupied. The reaction rate is controlled by the amount of substrate and, of course, the efficiency of the enzyme. In the body, the enzyme concentration can vary, and that's going to be dependent on what the metabolic needs are at the time. The body does not waste energy. It's very, very efficient. The reaction rate then, as I said, is controlled by the amount of substrate and the efficiency of the enzyme. And we see that the rate varies directly with the enzyme concentration as long as the substrate does not become a limitation. Let's take a look at some ways to increase the reaction rate. Remember, if we increase our temperature, that usually increases the rate of reactions, and that includes enzyme catalyzed reactions. But here's what's going to happen. As we increase the temperature, then the rate of the enzyme catalyzed reaction is going to increase first. It's going to go to a maximum, and then all of a sudden the rate is going to start to decrease. Why? Because biological enzymes are proteins, or they do have a protein part to them. And what happens to proteins? They will begin to denature at high temperatures. Extremes of pH will also denature enzymes. Most enzymes are going to have a maximum activity at a pH of about 5.9. I would not heat an enzyme any higher than 50 degrees Celsius. Because, again, you got to worry about it denaturing. As I said, the body's a very efficient machine. Enzymes must be able to turn off and on reactions, slow them down, speed them up. It just depends on what is needed. Okay, what does the body need? How does this work? Well, the activity of an enzyme is regulated. So let's do some definitions here. Any process that starts or increases the activity of an enzyme is activation. That makes sense. Activate the enzyme. Okay, that means you're going to increase the activity of the enzyme. A process that stops or slows the activity of an enzyme is inhibition. So we inhibit the enzyme. Feedback control is the regulation of an enzyme's activity by the products of a reaction that is later in the pathway. Oh, what do we mean by that? Well, many of our biochemical pathways are dependent on a series of consecutive reactions in which the product of one reaction is the reactant for the next. So here, this product B is the reactant for this reaction here, and this product is the reactant for this reaction here. So, for example, if B is not produced, then these two reactions are not going to take place. So the result of a process feeds information back to affect the beginning of the process. So that makes sense. I mean, think about our oven, right? Our oven is regulated by feedback. We have a sensor that detects the temperature and feeds back the information in order to turn the heating elements on or off depending on the temperature. So this is the same kind of thing. So in this case here, let's say product D inhibits enzyme 1. Then what's going to happen is the amount of A converted to B, or the amount of B, is going to decrease, and C will also decrease. So basically, with this type of feedback control, what's going to happen is the concentration, in this case of D, is regulated. So let's say in the body that more D is present than what is needed. Then what's going to happen is enzyme 1 is going to be inhibited. And finally, when all of D is used up and the body needs more D, then all of a sudden enzyme 1 is activated and D will be produced again. But in order for D to be produced, B and C also have to be produced. And we have to have available enzymes 1, 2, and 3. Okay? This is the body's way of conserving energy. It's conserved by inhibiting enzyme 1, and the B and C intermediates are not needed, so energy is not wasted. And as I said, your body is very efficient. Anyway, enzyme activity. We know it can be activated. We know it can be inhibited. So how does this happen? Well, the activity of an enzyme is controlled by the binding of an activator or inhibitor at a location other than the active site. So if this inhibitor or activator binds at a location other than at the active site, we call this allosteric control. An allosteric enzyme is an enzyme whose activity is controlled by the binding of an activator or an inhibitor at a location other than the active site. So the active site, what will happen is these enzymes, these allosteric enzymes, okay, can either activate the enzyme or, in other words, make the active site available by binding someplace else on the enzyme, or they can inhibit the enzyme. In other words, make the active site unavailable. Okay? So these types of enzymes have more than one protein chain and two types of binding sites. One of the binding sites, of course, is for the substrate, and one is for the regulator. The binding of the regulator, what it does is it changes the shape of the enzyme, and this in turn alters the shape of the active site. It either makes the active site available or unavailable for the reaction. So allosteric control can be either positive or it can be negative. Let's see what this means. So positive allosteric control. So we have this enzyme, and let's say the active site is not available. So a positive allosteric regulator, this molecule, right, it will bind to a site on the enzyme. It'll cause the enzyme to change shape, and the active site will become available so that the substrate can enter. The positive regulator is going to change the activity site in such a way that the enzyme becomes a better catalyst, and the reaction rate will increase. A negative regulator is going to change the activity site so that the enzyme becomes a less effective catalyst. And the reaction rate will either slow down or stop. So basically, again, we have this negative regulator. And, sometimes these can be drugs, right? We take prescribed drugs for different health issues. You could have a drug that acts as a negative regulator or a positive regulator. The negative regulator, so you have an active site, it's available, and so substrates are able to enter the active site. But with a negative allosteric regulator, it binds to the allosteric site on the enzyme. The enzyme changes shape in such a way as to hide that active site, and the substrate cannot enter. So we know about inhibition and we know about activation. And again, drugs are examples of substances that can either activate or inhibit an enzyme. It depends on the drug and on the health issue or disease that's being treated. Inhibition. So we're going to discuss now inhibition. Enzyme inhibition can be reversible or non-reversible. It can be competitive or non-competitive. So let's talk about reversible non-competitive inhibition. What does that mean to be non-competitive? What it means is the inhibitor, again, it could be a drug, does not compete with the substrate for the active site. So the inhibitor then would be an allosteric inhibitor. It exerts allosteric control by binding at a different site. So as we know, it would bind at a different site, change the shape of the enzyme, making the active site less accessible. Okay, so that makes sense. Going back to these allosteric enzymes, they can have more than one substrate binding site. And they can also have more than one regulated binding sites. There can be many interactions among them. So that means that very, very fine control can be achieved. That's the beauty of this. But anyway, back to this. So here is the reaction rate versus substrate concentration. And this is the curve with no inhibition. And here we're talking about uncompetitive inhibition. So again, that's where the inhibitor is not competing with the substrate for the active site. So this is allosteric control. So notice with the inhibitor, the reaction rate increases with increased substrate concentration more gradually. In other words, with no inhibitor, reaction rate increases pretty quickly. Here, we notice it increases very gradually. Okay. Also notice, I mean, we're always going to have a maximum rate, and that's because of the active sites being filled. But notice that the reaction, or the maximum rate, is definitely lowered. So the maximum reaction rate is lowered. It's not, this is way up here, and this is way down here. And once that maximum rate is reached, increasing the concentration of the substrate is not going to change the rate of the reaction. It's just not going to happen. Some of the statins that are used to lower levels of cholesterol and other fats in the blood. And, we know that they help prevent the problems that could be caused by cholesterol clogging the arteries. These medicines belong to a group of medicines that are called 3-hydroxy-3-methyl glutarate coenzyme A reductase inhibitors. And what they do is they work by blocking an enzyme that is needed by the body in order to make cholesterol. So because they block that enzyme, the body makes less cholesterol. Okay, and that could be good or in some cases bad. It just depends. Next, let's talk a little bit about reversible competitive inhibition. Then we'll go back to this chart here. So reversible competitive inhibition and competitive enzyme inhibition. This is enzyme regulation in which an inhibitor competes with a substrate for binding to the enzyme active site. So this is usually when molecules are similar in shape and size. What this means is a molecule other than the substrate can bind to the active site of the enzyme, which makes the enzyme unavailable for catalysis. Okay. Substrates in high concentration relative to the inhibitor can occupy more active sites. So if more inhibitor is present, then it will occupy more active sites. So it just depends. Is there more substrate? Is there more inhibitor? So it's going to depend on their relative concentration. So for example, methanol poisoning. Methanol, 100 milliliters of methanol will cause death. 25 milliliters will cause blindness. So how do they treat methanol poisoning? They actually treat it with ethanol. So they'll actually give the person alcohol, ethanol, in order to treat that poisoning. And the reason it's toxic is because methanol is oxidized to formaldehyde in the body. It's the formaldehyde that is dangerous. So ethanol, it's fairly similar to methanol in shape and size. So it acts as a competitive inhibitor of the enzyme methanol dehydrogenase. So methanol oxidation, that oxidation to formaldehyde, is blocked by the ethanol, and then the methanol is able to leave the body. So that's an example of reversible competitive inhibition. And again, whether the inhibitor or substrate binds to the active site is going to be dependent on their relative concentrations. Then we have irreversible, sorry about that, inhibition. And this is where the enzyme is deactivated, and this is due to the inhibitor forming covalent bonds to the active site, which permanently blocks the active site. No enzyme reaction can occur because the substrate is not able to connect to the active site. A lot of the irreversible inhibitors are poisons, and that's because they're able to shut down the active site. Mercury and lead are irreversible inhibitors because they form covalent bonds to sulfur atoms in the cysteine residues. Parathion, malathion are insecticides, and sarin, which is a nerve gas. These are irreversible inhibitors. And they're irreversible inhibitors of the enzyme acetylcholinesterase. What happens is they break down the chemical messenger acetylcholine that transmits nerve impulses. Without the enzyme activity, then the transmission of further nerve impulses are blocked. This is the nerve gas, by the way, resulting in paralysis of muscle fibers and death from respiratory failure. Again, as I said, most poisons are irreversible inhibitors. I'm going to stop right here, and that's pretty much it for enzymes. If you have any questions, please contact me. Everybody have a good day. Bye.